think it's okay. So, good morning, everybody. Rina, perhaps there is many wind. Many wind. Take, it's cold. So perhaps here or here. Here there is one here and there.
I'll go in the corner.
Because aircon is cold because at the back it doesn't work. So good morning, everybody. I hope that you had a nice evening and a good night. We are going to start. Some announcement. Yes, there is an announcement for our next meeting. Please come on resistance.
Thank you so much. I really appreciate it. So hi, everyone. Good morning. I just wanted to make you aware that we're going to have a azole-resistant international meeting. So I'm part of Rennes University. We already did a meeting last year. Some of you were there. And we are doing a follow-up, expand it a bit to make it a bit more global. We also have scholarships for PhD students. So we want to also make sure unrepresented groups can be part of it. I'll drop some flyers and you can check our website. And we got a grant to develop a knowledge repository for Aspergillus fumigatus resistant strains, but we'll cover from environmental to clinic and policy. We also work a lot with RIVM and other politic stuff. So yeah, it would be great if you want to join. Also a lot of data sharing and data management discussions. And if you have any questions, you can come to me. Yeah, thank you, and thank you for letting me.
Okay, thank you. And also you will see on the slides that we are organizing with David Denning the next Advance Against Aspergillosis and Mucorales. It will be at the end of January 2024 in Milano, in Italy. So yesterday we had a really brilliant day with very long day, but very impressive talks. But today it will be the same, and we start with Ester Segal. Ester Segal is professor of mycology in Israel, in Tel Aviv. Thank you, Esther, for being here. You know that it's a pleasure to work with you within the board of ECMM. You are the treasurer of ECMM, and it's really a pleasure to work with you, but not only work, to discuss and to do many things. So we are very, very happy to have you today. And please, you can start.
Thank you for the kind words. I really appreciate that.
Okay, thank you.
Okay. Well, so my name is Ester Segal, as you just heard it. And I am at Tel Aviv University. And the building that you are seeing here is the building of the in the campus. This is the preclinical building, the 7 or 8 departments. One of the departments is microbiology, where I am located on the 8th floor here. It's on the other side, so you cannot see that, but that's the building. The hospitals are not on, we don't have, it's not a good situation, but that's the situation. We don't have on-campus hospital, but we have 6 or 7, depending how it's counted, hospitals which are associated with the medical school. And the talk today is, as you can see here in this slide, there's a study on environmental fungi, of course, with focus on those fungi who are potential or known pathogens. So this is the topic, and the topic actually that I am going to discuss will be rare yeasts. And why rare yeasts? Because some of the yeasts we found in our studies are rare yeasts. Okay. Now, I past the slides where saying I have no conflict with anyone, including the people here. Well, so I'm not going to discuss guidelines, but I just brought this to show you that it is not such a rare thing that people somehow invented and say rare. This is really true. There are more common species, fungal species, fungal yeast, and there are many, many which are less common or rare. So there's a specific guideline for managing rare yeast infections, which you can see a large group. Many people of this audience took part, Rennes, for example, and others. So I will not be discussing that, but I am going to discuss environmental niches of some of these fungi. And what is the specific environment I am going to discuss is the following. The picture that you see here, actually on the right side, that I didn't take that picture, but this is a picture of Tel Aviv. Tel Aviv is on the coast of the Mediterranean Sea. And the data I will be showing are data from Israel, so because this is an international group of us that did many other beaches, but this particular lecture will concentrate on what was found in the Mediterranean Israeli coast. Okay, we had, as I mentioned, a survey going on for several years, international group, many people, including our chair. And what was done in that survey is that samples were collected from both sand and water and followed a specific soap procedure. Fungi were isolated and identified, like for other fungi, generally these days, Malditov mostly, and some also by sequencing, and were quantitated. But I'm not going into these details. I will only discuss yeast, which we found, but not the other aspects of the study. Okay, what did we see in the water and sand of the Israeli coast? There's 6 sampling sites, the whole coast. We had over 200 isolates. You can calculate it here. And you see that the majority, as could be expected, were molds. These were actually, so to say, a minority. And what also is obvious from this slide is that sand has much more fungi, so it must be a better medium for fungi than water. Maybe it's also the medium that we grow on, regular suborrow. Maybe it's not the best one because maybe some fungi cannot grow under these conditions. But the observation is, I think, valid because others found similar observations. Okay, so far, what have we learned? The 2 basic conclusions of this, that sand has more fungi than water, and that both sand and water have more molds than yeast. So these are the first observations, the first fact, so to say. Now let's see what kinds of yeast we have. This is what we found. I'm not very sure if you can read it from far away, but I will try to tell you. You can see we have rather a nice number of Candida species there, including Candida albicans. We have Parapsilosis, we have Tropicalis, and we have some rarer Candida. I won't be speaking about the other more common ones, but only the rare ones. But for example, we have Candida lipolytica, which is not very common, but it is known. Or Africana. And also we have some Cryptococcus species, I will mention that. We had Codamea, Rhodotorola species, and another Colariella. So, and what you can see, that sand, in sand we found all these species. Water, less. So maybe the conditions or some other factor which we don't know yet. Most of them are in sand. Okay, so it's an educational symposium, so I made it in the form as for students. So what is the conclusion? I know you are not at this stage, but that makes it simpler. Sand has more than water, and not only more, there are species which you cannot, or we could not, isolate from water, but only from sand. Maybe it's some technical, or it's real that because they are in smaller numbers, and that's why we couldn't isolate them. Now, regarding Candida, there's a wide spectrum of species, actually. But for the others, like Cryptococcus, there's less than Candida, but still we had several species, and one intriguing, I find it intriguing, was Cryptococcus uzbekistanensis, not so much known as others. And Codamea, I didn't know it before as well. Okay, so let us now speak a little bit about these rare yeasts that we found. Rare Candida, Africana, Lipolytica, Lusitania. Out of these, some of them are commensals, human or animals, but I will speak a little bit about Candida lipolytica, which is not associated with a certain host. From the Cryptococcus species, albidus is known, but uzbekistanensis again, and we found it several times, not only once. It's a rather rare species. And it's not very common, and so there are not too many patients, but there are some reports on clinical reports. So let's start with Candida lipolytica. It is an Ascomycetes yeast. It is sort of dimorphic because it can very well produce filaments. It is very common, and you can have it in meat and cheeses. And one of the reasons why it's used as in food industry is because it has an aroma, special aroma. So if you have a very tasty cheese, maybe you eat it. That's what they say. What about us? Because we are the focus of everything in there, aren't we? So it is a rare, of course, but there are enough cases to be important, so people have to know how to identify it, and these are basically the regular methods used, nothing special. Now, in terms of treatment, voriconazole, decanocanidins, and amphotericin B. And if you are really interested, that's where you can read all the details. Okay. What about something more about this yeast? It is also can be found, and it's described in children. So it's not only in adult mostly with some background, but in children as well. There are it's quite relatively known already for 20 years, so it's nothing really very new. But I don't know if every clinician or is aware of this. What would you say?
Well, all the candida names have changed, so I think everybody goes back to the books and Google immediately when you report something. Sorry. Yeah, my comment is only about when you get a new candida name, a new candida species, my first thought is that, oh, is it an old one? I just know the name. Names have changed.
Okay, so and it not only is it's known, it is also outbreak. There are outbreaks. So if whoever is in a hospital environment might really hear and see this species. And it's not only, as the slide says, it's not only in the past, it's here. It's here, and as expected, if it's here, it will be also associated with corona. So there are cases, as you can see, very recent case as a plus to the corona. Now, I won't go into other candida species. We don't have the time for all this. And I'm moving to Cryptococcus. Now, I'm not going to describe the regular cryptococci, which I'm sure in this audience everyone knows. But I want to say a few words about Cryptococcus uzbekistanensis. Albidus, of course, is also known, but albicans is rare, albidus is rare, but not so rare, actually. If you look in the literature, you can see, do you see in your hospitals albidus?
We had one recently.
One. So it's relatively rare, but not as so rare. Now, what is really rare is the Cryptococcus, now that's called Naganitia. This was the name, I heard it from from CBS, so it's officially Naganitia. And that's Basidium acetous yeast. It can be found in patients with AIDS, in leukemia, lymphoma, and other debilitating conditions, probably also. And I would like to speak a little bit about the Uzbekistanensis, because this appeared, I don't have no good reason why, in two different, we explored 6 beaches around of the coast, and two beaches which are close to the southern part of Israel, and it's not far from a port, from one of the 2 major ports that are on the coast. Okay, so this is the one, Uzbekistanensis, Naganisha Uzbekistanensis. It was first described years ago, some years ago. It's not in a regular journal. It was an army report, American Army report. It was found not 6. So from what I could find out, not from 6 soldiers, but it was somehow also, because this is not published in regular journal that I would have or you would have access to, but in military reports from the Middle East, American military reports. So it was known, but it's a very rare species. And just by the name, you can also see it's not very common. And it was not known to cause disease, except for a few years ago, there was a report in the US, but in an older man in California who immigrated from the Middle East. Now this fungus is the CBS. It's deposited there. It is sequenced and has a new CBS number. I don't know by heart, but it has. If you want, you can have it from there. But it's really very rare. And this is the case I was mentioning before, about 10 years actually already, that was isolated from an immunocompromised patient with lymphoma in California. But someone who immigrated from the Middle East. Okay, everyone knows these fungi. Rhodotorola, very, very pretty to look at. And the 2 common species are Rhodotora glutenis, and you can see even the Hebrew letters behind that the student wrote. And we also had some other rare ones, the Codomea. These 2 species we found, and Codomea is, of course, also quite rare. How many Codomeas do you have? 1. Anyone else? No one. Okay. So it's really rare. What can the Rhodotorola do? A lot, actually. The main pathogens are the human pathogens, 3. There are more of them, but others are even rarer. Mucilaginosa, glutenis, and minuta. And you can identify them, not so difficult to identify. They are Basidiomycetes yeast, and where are they found? In soil, you can read it, ocean and lake water. So we found it in the ocean, the sea, Mediterranean Sea. But it is also, you can see it's, you can find it in juice and milk, or at your own home on a toothbrush that if you don't change it frequently, or on the shower curtains. So actually all over. Now, Rhodotorula is an important pathogen, actually. And it's associated with modern medicine. So I am pretty sure that anyone who has some association with a laboratory in a hospital would see it. I will try to be faster. Okay. Now, diagnosis, like in others, the regular diagnosis. And treatment, sometimes you can, you don't need even a treatment, but if you do, it's amphotericin B and 5-FC. Codomea, I'm trying to be faster. Codomea is an Ascomycetes, and it's quite, actually, although the name seems not so known, but it's quite common, so to say. Here, some data about Codomea, cases of fungemia, endocarditis, and other infections in pediatrics, but not only in pediatrics. And you can see the treatment is basically amphotericin B. So, and I have still some, if I will have time, I will cover these 4, but if not, I can stop any minute.
Just a few minutes now. A few minutes.
A few minutes.
4 or 5 minutes.
Okay. Trichosporon, very well known. It's a Basidiomycetes yeast. You can see it, very typical colonies. Ecology is very interesting. It has specific ecological niches, and it is associated with humans and animals. These are the common 6 species. I think a very nice reference on this you can read from Maiken, right? Okay. And the diseases it can cause, some of them are superficial, not very common diseases in Europe, but there are many, many cases in poor areas in the Orient and in Africa, where the hygiene conditions are poor. Laboratory diagnosis, nothing special. These are the treatments for these infections. But these are species that can also cause severe systemic infections, like disseminated infections in compromised hosts, even in AIDS, which is also a compromised disease. Okay, so in immunosuppressed patients, I already said, but I want to add to it also eye infections. That's an important yeast for eye infections. I knew the name Geotrichum. Now it's called Magnesiomyces. So it is an Ascomycetus yeast, and I don't have the time to go into all these details. But it's an important, rather not so common yeast. And what does it do? Many, many things, as you can also read here. And one more. I'm trying to be fast. And one more again. And that's the end of it.
So thank you, Esther. Yes, as you know, we are online with many,
Yes.
many people, so we have to
That's
the
fine.
time.
That's fine.
Yeah.
No, don't worry.
We're increasingly reading reports about the isolation of Candida auris from wastewater treatment plants, both in Europe and in the US. And quite often, the effluent from these wastewater treatment plants is discharged into the sea. Now, I'm not suggesting I've been there. The beautiful beach at Tel Aviv is contaminated with auris, but you didn't mention auris. Are you aware of auris being isolated from sand and water?
Well, the question, a very good question. You know Daniel Elad, I am sure. This is a colleague of mine in Israel. He didn't publish it, but he did a survey for specifically looking for auris, but he couldn't find one. Okay. But there are quite a number of cases. So the first, actually, auris cases were imported to Israel from South Africa. Someone who visited South Africa came back with auris, and then it spread like in other places.
And as you know, of course, Anuradha Chaudhary showed that Candida auris is in sands in South India. So thank you very much, and we move to the next presentation. So I'm happy to welcome Sandrine Giraud. She is professor in Angers University, not so far from Rennes, and she will talk on rare filamentous. She's a specialist on Schistosporium.
Thank you very much, Jean-Pierre. Good morning, everyone. So, as you know, or as it was said by Jean-Pierre, in the lab we are interested in the fungal respiratory infection in the cystic fibrosis. But I will not talk about this because this will be the topic of another talk later. And so one of the questions raises one of the questions deals with the origin of the contamination of the patients. And this led me to take attention to the ecology of some human fungal pathogens and to their environmental reservoir. So as it was kindly offered to me, I would like to present you an overview of the environmental reservoir of some rare filamentous fungi pathogenic for human. Next, it's here, so I have to put, yes, good. So the airway exposure to environmental smog is constant, you know, and very few fungal species are able to cause disease in human. Of the estimated 3 million fungal species, only 300 can cause human disease. So what makes a successful pathogen? The first thing, one key requirement is that they must survive at elevated temperature. You know, most fungi grow best at ambient temperature, and the human body temperature is too high for them. But growth at 37 degrees is not enough, of course, and most of the filamentous fungi may cause disease in host with predisposed factor like a weakened immune system. For the pathogen, the host environment is quite harsh with a wide variety of oxygen stresses and other attacks by the host immune cells. And the defense mechanism It's similar to those, the defense mechanism is similar to those developed by fungi in response to the stress they encounter in their environmental niches. And this suggests that their virulence factors have evolved in response to threats to which they are exhibit in their natural niches. So to be efficient, again, fungal infection or fungal colonization, it is important to understand the mechanism of selection in the airway of the mold and their clinical significance in the clinical significance of this isolation. It is also important to better understand the pathogenic mechanism in order to develop new and more efficient therapeutic approaches. However, study on ecology of these fungi may provide crucial information about the origin of the contamination of the patients and may help us to define prophylactic measures aiming to reduce the environmental exposure and, of course, the risk of for contamination. So, most fungi are free-living heteroprophyte saprophyte due to their high plasticity and due to their capacity to develop various forms in response to unfavorable conditions. So they are very successful inhabitant of the soil. They are able to produce a wide variety of enzymes, and so they can degrade soil compounds, and thereby they regulate the balance of carbon and nutrients. They convert the solid organic waste into biomasses, carbon dioxide, and also organic acids. So as soil is the natural habitat of various fungal species, including opportunist pathogens belonging to Aspergillus, Fusarium, but also Pseudosporium species, gardening activity is a major circumstance of contamination. I will talk also about compost, like yesterday. As they are able to use various carbon sources and they are able to survive in extreme environment, fungi may play an important role in the composting process. You know that organic waste have become an environmental concern, and composting is one of the most efficient method for substantial waste management. So this complex process depends on various factors, like the resident microbial community, the nature of the substrate, its nature, its composition, and also its size, and physicochemical conditions like the temperature or the oxygen content. So usually, composting process entails 3 different phases, characterizing by different microbial communities. So first, during the mesophilic phases up to 40 degrees, the soluble and readily degradable component of the compost are digested by the mesophilic microorganism. Then there is an increase in temperature over 40 degrees during the thermophilic phase, and this allows the selection of thermotolerant and extremotolerant fungi adapted to high temperature and also essential for a biochemical process, like, for example, lignin degradation. And then the mesophilic microorganism take over during the curing phase and participate to the maturation of the remaining organic content. The ratio between fungal and bacteria in the compost is about 2.1. This means 2 fungi for 1 bacteria. Okay, so during the last decade, several authors studied microbiota in compost, and a very large number of potentially harmful fungal species were found. So Aspergillus fumigatus, Aspergillus terreus, and Chelosporium apiospermum were recovered during the whole process and at the end in an active composting plant. Pseudosporium aurantiacum, Pseudosporium deugai, but also Rasamsonia emersoni were identified as fungal species associated in compost. Lomantospora prolificans was isolated or is present in the maturation phase of tomato plant compost and is present at the end of the process. And Aspergillus fumigatus and Pseudosporium boydii were described as the most frequent species in compost, in green compost especially. In addition to the proliferation of these thermotolerant or extremophilic fungi, the composting process constitute a risk for human health, especially for people that are living close to, in close proximity to the compost facilities, since bioaerosol containing biological hazard like fungi or bacteria may be generating during the process. And Aspergillus fumigatus was regularly reported as one of the most abundant fungal species in the bioaerosol. Other investigation on thermophilic fungi, on different type of compost like municipal, agricultural, or horticultural, evidence a high occurrence of Rhamsonia emersoni in the air. So one important parameter in this case is the distance at which the bioaerosol remain detectable. And on this subject, in the literature, you can find a very high different results from 30 meters to 1,400 meters. And as it was said yesterday, you can also find in the compost the presence of azole resistance among filamentous proliferating in this environment. It was said yesterday also, but green waste, wood chipping, and flower bulb waste were the first hotspot identified for azole resistance in Aspergillus fumigatus. And well, how we define azole resistance Aspergillus fumigatus hotspots? It's an environment containing azole fungicide, of course, and providing favorable conditions for fungal growth and spread for a prolonged period. The environmental reservoir to which any individual may be exposed result from all their activities. So this means professional, also the hobbies or the school, these kind of things. And you know that in the majority of time is spent in indoor environment. Okay, so this means that indoor air quality may constitute a risk of being exposed to environmental mood. In indoor environment, humidity is the major factor of mold growth, either visible on painting wall or on wallpapers, or either invisible. And here you can see a table representing the most level required for fungal growth in construction or in finishing or furnishing materials. And as you can see, some Aspergillus and Exophiala species were reported as primary, secondary, or tertiary colonizer. So, alongside this environmental reservoir common to various species, fungal species, some environmental study highlighted some species-specific environments. So, I will not now continue on the ecology of three filamentous fungi, of course, Pseudosporium, yes, Exophiala, and also Fusarium a little bit at the end. So, Pseudosporium. Among the 15 species known in the Pseudosporium genus, only 6 are reported in clinical setting, and only 5 really reported in the as colonizer of cystic fibrosis patient: Pseudosporium aurantiacum, Pseudosporium apiospermum, Pseudosporium boydii, Pseudosporium minutisporum, and Pseudosporium ellipsoideum. Pseudosporium species are soil saprophyte, and they are mainly encountered in temperate climates and less frequently isolated in tropical area. And they exhibit particular properties that explain that they may survive in extreme conditions. So they are able to survive at very low oxygen pressure. They tolerate high salt concentration and high osmotic pressure, and they are able to use natural gas and to degrade aliphatic and aromatic compounds and use this as carbon or energy sources. Study on ecology of Pseudosporium species was clarified owing the development of semi-selective culture medium like the Scusalpus medium or the Pseudoselect3 medium, and several environmental studies were performed in Europe, of course, but also in Thailand, in Morocco, in Australia, or in China. And in this study, Pseudosporium species were never recovered from natural habitat like forest in Australia or in France or seashore in Morocco and in the Netherlands. And Pseudosporium species were mainly isolated from anthropized environment like polluted area or wastewater treatment plants. Pseudosporium species were firstly isolated inside building in 1989 from 5 potted plants in a corridor of medical unit in Toronto Hospital. Of note, these potted plants were revealed also to be positive for culture positive for Aspergillus fumigatus and Lomantospora prolificans. In another study performed by Began and Nolar, Pseudosporium species were never recovered from the air or surface sample collected in 130 dwellings in Belgium. In a study performed in the lab, an environmental study conducted at home of 6 patients with cystic fibrosis and colonized by Pseudosporium, we collect a total number of 136 samples in bedroom, in living room, in bathroom, including hair, water, surfaces, but also soil sample collected from garden or soil sample collected from the potted plant, all the potted plant at the home of the patients. And we are not able to identify Pseudosporium, to find Pseudosporium species in the air, in the water, or in the surface sample, except for one, in the heat radiator in the bedroom of one patient. The samples collected from pet litter or the soil sample collected from garden were also culture negative for Pseudosporium. And what was very interesting is that 70% of the sample collected from potted plants were positive for Pseudosporium. So this study clearly identified potted plant as in the reservoir of Pseudosporium species. In this study, it was also performed a correlation, a comparison of the genotype of this environmental isolate to the genotype of clinical isolate obtained from sputum sample of patients. And you can see here one of these patients, the result of the genotype of the clinical sample and the genotype of the environmental sample. As you can see, there is a huge variety of genotype in environmental sample, but just one unique genotype in the clinical samples. And what is very interesting is that this genotype is similar to 3 genotypes encountered in the environment, as you can see here. You have the same phenotype as the genotype from the patient. So the potted plant constitute really a risk factor for airway contamination of the patients. Exophiala dermatitis is also common in the cystic fibrosis in some countries, like in Germany or in Sweden. And this is a polyextremotolerant fungus able to grow in a wide range of temperature and a wide range of pH. Exophiala dermatitis are highly tolerant to salts, up to 3 molar NaCl, for example. They are able to assimilate aromatic hydrocarbon, and this property may explain why some detergents are used as enrichment substrate for Exophiala dermatitis. And due to the high content of melanin, Exophiala dermatitis tolerate high doses of UV and ionizing radiation. Although Exophiala dermatitis is regularly reported in the clinical setting in some countries, its natural inhabitat is unknown, and it was just sporadically isolated in environmental samples. So it was found it was isolated from plants from the cuticle of leaf-cutting ants in Brazil, and also from tropical fruits and faces of frugivorous birds, hornbill and flying fosses in Thailand. So all these results, based on all these results, tropical rainforest is thought to be the main natural source for Exophyla dermatitidis. Apart from its natural niches, Exophyla dermatitidis is associated with a polluted environment rich in hydrocarbon, as suggested, for example, for Chelosporium apiospermum. A high frequency of Exophyla dermatitidis was found in creosote-treated railway sleepers in Turkey, but also in Iran. And Exophyla dermatitidis was also found in crude oil in Britain and in a sample collected from some petrol station in Slovenia. The isolation of Exophyla dermatitidis was also reported from water or water-related environment like deep sea sediment in India, or beach sand in Greece, or glacier in Italy, in Slovenia and Antarctica. And all these results suggest a possible connection with low nutrient water. Considering the indoor reservoir, Exophyla dermatitidis was isolated first from niches in regular contact with water, like sludge from bathroom drain pipes and kitchen sinks. The occurrence of Exophyla dermatitidis is associated with high temperature and the presence of water. So steam baths, reported as an indoor reservoir of Exophyla dermatitidis, combine the high temperature, the presence of water, in addition with alkaline pH. Moreover, dishwasher are insidious reservoir for the human filamentous fungi, especially Exophyla dermatitidis. And the Exophyla dermatitidis was regularly detected on plastic element in the dishwashers, silicone and rubber seals, due to their, since the rugosity or the hydrophobicity favor the development of biofilms. Exophyla dermatitidis was also regularly reported from environment related to dishwashers. This means that the wastewater sample or the hot aerosol or the surface kitchen with the dishwashers. And it was also reported in biofilm at domestic water tapes and in groundwater samples. So this result suggests that the tap water system represents a connection between the indoor environment and the natural water environment. Just a few words to finish with Fusarium species. So, you know, the Fusarium genus comprises high diversity of species, which are primarily plant pathogen with a worldwide distribution. They are a major constraint in food security, not only as pathogen of plant growth, but also as producer of mycotoxin-contaminated chain food. About 70 species group into species complex Fusarium species, so are able to cause infection in human, but most infection in human are caused by only 4 species complex, and these 4 complex are cited here, Fusarium solani, for example, or Fusarium oxysporum species complex. Okay. Most of these species are readily isolated from environmental sample, including soil, plant material, air, aquatic environment like river and seawater. In a study performed in an hospital in Houston and published in 2001, the water distribution was for the first time reported at the indoor reservoir of Fusarium, and Fusarium species were isolated from sample collected from drain, faucet, shower heads. These results were confirmed later in several studies, showing the presence of Fusarium species in hospital, but also in domestic or in municipal water systems. In this study, an aerosolization of Fusarium species after running shower was suggested, since Fusarium conidia was found in the air, isolated in the air. And finally, a molecular match between patients and environment sample was observed in this study for 2 patients, and the 2 patients are here, here, and here. So this suggests that the patient may be colonized, may be contaminated by their environment. Okay, I hope I convinced you that it's important to study the ecology of this fungi, and thank you very much for your attention.
very much. Thank you very much, Sandrine. Do you have some question? Yes.
It's safer to wash by hand.
It's better.
So hi, thank you so much for your talk. And I was just thinking that so nowadays, basically, we cannot have plants at home. We must change curtains. We must change the shower, the pipes, the water pipes. And I remember this case of this kid in the UK that died of environmental mold pollution in his own house. And I'm wondering at which point can we demand that then the houses we live in should be checked for this?
What is important to keep in mind is that these mold are dangerous for people with an immunocompromised system. So for most of people, there is no problem to have all this kind of thing. But for people that have predisposing factors like an immune suppression or things like this, I think you have to take care of. But of course, you can avoid everything, but you have to take care of some point and eliminate some point of contamination.
Yeah, for example, during the COVID, right?
Yes?
You have the microphone there, please.
Well, so during the COVID, for example, we were all kind of in a higher risk of to have our immune system lower. So at which point can we really demand? I'm just trying to understand in terms of society, what can we also demand to be sure we are a bit...
You have to stay in a sterile place.
Yeah.
No, I know.
But it's then an issue, right? There's still really no plan for when this gets worse. What can we...
You have to find the balance between the risk and what you want to do.
May I add a comment?
Wait, with the microphone, because we are online.
You know, if you have a child that is allergic to something, like dust allergy, then it's, for example, not recommended to have curtains or some kind of carpet. So there are some. It's not a I think it's some sort of a regulation, but if you take your child to an allergologist, voila. Then he will advise you, don't put curtains, take out from the room where the child sleeps the carpet and so on.
This would be the topic for this afternoon, okay? Monitoring and then remediation. So we will have different talks on that. Yes, one more
Just
question.
a very
Rina?
quick reply to you about the UK case. I mean, if you ever worked with a mouse model, you know that you can expose the mouse to a little bit of chlamydia or something, and you may get a bit of an airway disease. But you give it enough chlamydia, they get invasive disease. So the same applies here. This kid lived in a moldy house that looked like it had been in the bottom of the sea for a while. It was absolutely black and moldy. You know, we're talking about extremes here. Not your house, I mean, seriously.
I'm just wondering to what point is someone paying attention to
Yeah, so exactly, and that is what we are working on, and that's today the afternoon session about, monitoring what's needed and where do the cutoffs, if you say that there is a cutoff, where does it come from? Yeah. Where's the evidence?
Don't worry, we talk on that in hospitals, at home, and at work, because these three situations are at risk. Thank you very much, Sandrine. So now we move to the next one. Professor Gilles Nevez is Professor of Medical Mycology in Brest Teaching Hospital in Brittany. And he will talk on a very special fungi, as Pneumocystis cerevisiae.
My slide.
Okay. Here you can see another part of Brittany, very nice part. Thank you, Gilles.
Thank you, Jean-Pierre, for organizing this sympo and inviting me. It's very kind. I would like to add that I am a member of a research team called Fungal Respiratory Infection. It's a research team localized in two universities, Brest and Angers, and this team is directed by Professor Nicolas Papon. Nicolas Papon had a talk yesterday on the secondary metabolites. I also the head of the laboratory of mycology of the University of Brest. Brest is located there in Brittany, 250 kilometers from Rennes and 600 kilometers from Paris. So why going on working on pneumocystis and pneumocystosis? Because in fact, infection due to Pneumocystis jirovecii are still a public health issue. This slide presents a short, non-exhaustive historical review. And you know that in 1912, the Delanoës, Mr. and Mrs. in Paris, they identified this in guinea pigs and rats, and they call this microorganism Pneumocystis carinii. But they also showed the transmission between female rats to the neonates, and this important finding are usually omitted. Until the 80s, the microorganism was essentially reported as a pathogen in children and a preterm infant. But recently, a Spanish team shows that the microorganism was still involved in respiratory distress syndromes in preterm newborn infants, so in Europe, specifically in Spain, and also in France. Something happened in 1981. This is a dedication to Sandy Ford. This dedication was published in Emerging Infectious Diseases in 2016. Sorry. Who was Sandy Ford? Sandy Ford was a sort of whistleblower, the technician of the CDC in Atlanta, who notified the federal authorities to a new epidemic in the USA, given to the dramatic increase of pentamidine use for treating atypical PCP cases in gay men. Also in 1981, later in 1981, you know that AIDS was described and defined by two American teams, Gottlieb and colleagues in LA, and Henry Mazer and colleagues in New York City, through the description of these atypical cases of PCP in AIDS patients. In gay men. During the 80s and until the mid-90s, there was an important relationship between PCP and AIDS. You know that. And in the mid-90s, the use of antiretroviral therapy for treating HIV infection in developed countries led to an important decrease in the frequency of PCP in AIDS. However, currently in metropolitan France, PCP remain the most frequent AIDS defining diseases, with about 30% of AIDS cases related to PCP. And also in France, PCP in HIV negative immunosuppressed patients represent about 70% of the overall PCP cases, so a number higher than the number of PCP in AIDS. In other respect, about 400,000 PCP cases may occur per year worldwide, with a mortality rate up to 18%, considering the study by Brown and colleagues. After several workshops on pneumocystis, it was admitted that there were different species in the Pneumocystis genus, and we decided to call Pneumocystis in human Pneumocystis girovesi in honor to Professor Otto Giraud, a Czech researcher, Czech pediatrician, sorry, as suggested by Jakob Frankel. And we kept Pneumocystis carinii to call Pneumocystis only in rats. So there are different species in the Pneumocystis genus. 5 species today are correctly identified: Carinii and Wakefieldii in rats, Murina in mice, Orytolagi in rabbit, and Girovesi in humans. But there are probably other species. For example, in dogs, maybe you know that Cavalier King Charles are very susceptible to Pneumocystis. And also, probably some species in monkey population, for example, in macaca. Jacques Guyot worked on this topic many years ago on Pneumocystis in macaca population. So there is a host specificity of Pneumocystis species, and an animal reservoir for Pneumocystis girovesi can be excluded, and the specific mammalian host infected with Pneumocystis may serve as its own reservoir. And pneumocystosis in humans is an anthropognosis with human as a reservoir for Pneumocystis girovesi, and individuals infected with Pneumocystis girovesi may represent putative infectious sources. And this, if we consider that there is no ecological niche for Pneumocystis in the environment. And if you consider that there is no exosaprophytic form of Pneumocystis. What about the outdoor detection of Pneumocystis? Pneumocystis DNA has been detected in 1 sample of pond water, and this was fascinating. This was relevant since you know that Pneumocystis, in fact, lives into the alveolus lumen and in the surfactant, which is a liquid medium. But this meeting report was not further published, and this topic was not investigated later. The paper by Anne Wakefield is more interesting. In this study, Pneumocystis DNA, Pneumocystis carinii DNA, Pneumocystis girovesi DNA was detected in air sample from apple orchard in Oxford. And this paper was frequently referred by some teams as a proof of the existence of the environmental reservoir. But it is not so simple. And Anne Wakefield and James Stringer, in a co-author paper, wrote about this information paper by Anne Wakefield, that in fact, in the number of Pneumocystis organisms seems to be very low, and Pneumocystis carinii and Pneumocystis girovesi may have been deposited by animals and humans, and they in fact assume the probable absence of the environmental reservoir for Pneumocystis species. In fact, the sole place where Pneumocystis has been visualized are the infected mammal organs, and mainly the alveolus lumen. But Pneumocystis presence in the air, in the circumstances of exhalation, transmission, or inhalation acquisition, can be investigated. They were investigated. The contribution of rodent models to PCP knowledge is major, and the airborne transmission of Pneumocystis carinii in rat was investigated and proved by Hughes and colleagues in the 80s. Sorry. They use a germ-free rat model. And the germ-free rats, immunocompromised with dexamethasone, when maintained into an isolator with filtered air, do not develop PCP despite their immune status. Conversely, if these germ-free rats immunocompromised with dexamethasone are exposed to a conventional rat into the isolator, once again, an isolator with filtered air, the germ-free rat immunocompromised develop PCP. And it is noteworthy that in this experiment, there were no direct contact between the rats. The contacts only took place through the air into the isolator. The airborne spread of Pneumocystis carinii from rats were studied by Choukri and colleagues. They use an isolator with a nude rat, an isolator with a filter, and they use a the Coriolis micro air sampler to take air sample from the isolator, and they use a qPCR assay to detect and to quantify pneumocystis. And they found a significant correlation between burdens of pneumocystis in air sample and in the lungs of rats. And this finding showed that Pneumocystis carinii was effectively exhaled by the infected new rats. Pneumocystis murina is a highly transmissible microorganism in mice, and Pneumocystis murina can be transmitted from immunosuppressed kid mice with PCP to immunocompetent biopsy mice which do not further develop PCP, but these immunocompetent biopsy mice can be colonized by pneumocystis. And these immunocompetent colonized biopsy mice can transmit Pneumocystis murina to other skid mice who further develop PCP. And this model, the skid to biopsy mouse transmission model, was developed in Lille by Anne Dumoulin and Magali Chabet in the leadership, on the leadership of Dr. Eduardo Deicas. Pneumocystis cerevisiae transmission was suspected through the occurrence of clusters of PCP cases in hospitals. And this table shows the main cluster described since the advent of molecular biology. This is not an exhaustive review. And in the studies indicated there in this table in shades of gray, the analysis of, sorry. It was not a joke. So, in these studies, the analysis of the encounters between patients, encounters between possible index patient and possible contact patient, and the result of pneumocystis genotyping were compatible with patient-to-patient transmission. As you note, there is something interesting concerning the underlying condition of the patient. Most of the underlying condition of the patient was organ transplantation. So in 2020, we investigated pneumocystis infection outbreak in organ transplantation unit in France in a collaboration with our association, the Anophel Association in France. It was a survey, and during the survey, we noticed that over 12 years, 16 outbreaks occurred, including 13 among renal transplant recipients. And we are now working on the relationship between pneumocystis, outbreak, and transplantation, but maybe it's a little bit off topic today during this course, a CMM course. In fact, none of the studies dedicated in this table, none of the studies have investigated the role of patients with pulmonary colonization with pneumocystis. So we worked on this topic, and we provide molecular evidence of colonized patients as potential infectious sources of Pneumocystis cerevisiae in a context of a cluster of pneumocystis infection among renal transplant recipient clusters that we were confronted to. In this study, 10 patients were considered as possible index, of whom 7 presented PCP, but 3 were colonized by pneumocystis. I think that, in fact, investigation the indoor Pneumocystis cerevisiae in the hospital premises, it's a good approach to investigate the airborne spread of Pneumocystis cerevisiae from PCP patients. In fact, Pneumocystis cerevisiae can be exhaled by infected patients in their surrounding air. In this paper by Matt Olson, Pneumocystis cerevisiae DNA was detected in air filters in rooms of PCP patients and in other hospital premises. And they conclude to the possibility of acquisition of pneumocystis from the environment. Why not? Then we can say also to the possibility of pneumocystis exhalation by infected patients in the hospital. It's a paper that was published in 1988. The study by Singh and colleagues is more interesting. Pneumocystis cerevisiae DNA was detected in an intubation system of a PCP patient, but upstream of the respirator. And this is a very important paper. It showed that pneumocystis can be effectively exhaled by an infected patient. It's a major paper. The study by Maher, Sandy Maher, and colleagues is also very interesting. In this study, pneumocystis cDNA was detected in air filters in rooms of HIV-infected patients with PCP. And this study shows that, in fact, suggests that viable pneumocystis organism can be spread by infected patients in the hospital environment. Okay. The paper by Choukri and colleagues is fascinating. Pneumocystis DNA was detected and quantified in air surrounding PCP patients. They use the Coriolis microair sampler to take air sample in the surrounding air, or surrounding air in the room of PCP patient. They use a PCR assay to detect and to quantify pneumocystis cerevisiae. And in this study, at 1 meter distance from patient, pneumocystis DNA was detected in the air sample in 15 out of 19 PCP patients. Pneumocystis cerevisiae DNA was also detected for some patients at 3 meters, 5 meters, and 8 meters. And the level of P. cerevisiae DNA decreases with the distance from the patient. And this result suggests or shows that pneumocystis organism can be exhaled by infected patients in their surrounding air in the hospital. Using the same method, we investigated pneumocystis cerevisiae in the air surrounding patients with pneumocystis pulmonary colonization. And pneumocystis cerevisiae DNA was detected in 5 out of 10 air samples collected at 1 meter, and also in 5 out of 10 other air samples collected at 5 meters. And all of these data, in fact, suggest or show that exhalation and potential airborne transmission of pneumocystis is possible from PCP patients and colonized patients. What are the other potential human sources of Pneumocystis jirovecii in the hospital? We know that immunonaive infants develop pneumocystis primary infection early in life. Pneumocystis primary infection can be asymptomatic or symptomatic, and in symptomatic infant, in symptomatic primary infected infant, Pneumocystis jirovecii can be detected in nasopharyngeal aspirate with a peak between the 3rd and the 6th month. So we investigated the pneumocystis exhalation by infants developing pneumocystis primary infection. We examined 14 infants, and we detected pneumocystis DNA in air sample in the room of the infant in 3 air samples with a perfect genotype match in one couple of air sample and NPA. And these first results are compatible with pneumocystis exhalation from infant with primary infection. I wonder if these results concerning the indoor pneumocystis detection in hospital reflect the indoor household or outdoor pneumocystis circulation, that is to say, in the community. I think they do, probable. What is the pneumocystis stage that may correspond to pneumocystis DNA detected in exhale air sample, considering that the exhale stage is the same as the transmitted stage and the same as the inhaled stage. In fact, Melanie Cushion and colleagues show that mice treated by echinocandin for PCP do not harbor pneumocystis cyst, today ASCII. And these mice can harbor trophic forms. And these mice harboring trophic forms but do not harbor, not harboring pneumocystis ASCII cannot transmit pneumocystis to susceptible mice. And they conclude to strong evidence for the cysts as the agent of transmission. We can be more subtle, saying that the ascus, the cyst, formation is necessary for pneumocystis transmission. To conclude, may I ask you a question? Considering that Pneumocystis jirovecii is the species that specifically infects humans, that it is not routinely cultivable, that it does not have a proven environmental niche, that is transmitted through the air from host to host, and it shows a strong tropism for the lungs, do you think that it's really constitute a unique phenomenon in medical mycology that I wrote in this note in Trends in Microbiology? That's all. Thank you.
So, thank you very much, Gilles. Do you have some question on pneumocystis and pneumocystosis? Yes, Laurence.
Thank you, Gilles, for this night talk, and thank you for mentioning Eduardo Deica's work. I wonder, you talk about the importance of transmission between colonized and infected patients. How do you classify your patients or your colonized patient? Infected versus colonized.
What is the definition of a colonized patient?
No. Yeah. How in practice? Because the difference between the two cases, the two status, are very are not so easy to do. So what is your way to classify patients?
In the study published by Solène Legal in Clinical Infectious Disease concerning the transmission of pneumocystis from colonized patients, in fact, the definition of these colonized patients in these studies was the absence of microscopic detection in the lungs of this patient, first, and the fact that the fungus was only detected by using a qPCR assay, and of course the fact that the clinician, the physician, decided to not treat this patient. I think that's the first definition of probably a colonized patient. May I answer I answered your question, or yes, no? Yeah. Okay.
Rennes?
Yeah, just a brief comment about the. I think it's fascinating. Yes, definitely, it is a patient-to-patient transmission disease, which is actually very rare in mycology. You think you have your dermatophytes, but even Candida in normal environment, you know, Aspergillus, nah. You know, you have your own Candida colonization, and Aspergillus comes from the, you know, the nature, whatever. So it is a very special fungus. I think far more important is from infection prevention and control point of view. Do you need to put these patients into isolation rooms or isolate from other patients at risk?
Yeah, we work on this topic. We work on the exhalation of patients treated by sulfamethoxazole, and we learned, we investigated the degrees of the exhalation of pneumocystis from patients treated by sulfamethoxazole in order to define the duration of the isolation of PCP patients.
Or is it individual, like with TB, when they are staining negative?
Exactly. In fact, it's a fungus, but think about tuberculosis.
I have a technical question.
Oh, gosh.
Not really technical, but if, for example, France is a big country, the biggest in Europe. That's what I heard. And if there is a patient in Alsace, let's say, are you a center? Will he or she be transferred, or the samples will be sent to you?
To speak about reference center for sending pneumocystis isolate. Officially in France, we have a reference center, which is in Paris at the Pasteur Institute. But we are also a sort of reference center to analyze eventually your pneumocystis isolate. Yeah.
But usually in all teaching hospital, pneumocystis PCR is performed, beta-D-glucan is performed. So for the management, for the routine management of the patient, then it's quite easy in each hospitals. But in case of investigation of epidemics and special situations, then the team in Brest can be at disposal.
Yes, of course.
Your turn.
Okay,
It will be free.
Yes, but some publication and and some nice talks. So thank you very much, and now it's the coffee break, and let's meet at 11 after. Thank you.
Alors, je parlais pas.